Good morning. Let us continue with DNA replication with the machinery and the enzyme. Now the question is, who is going to provide energy for this entire replication process? Because when we say the enzyme is going to create a new strand altogether, creating new strands are like polymerization reaction. Means joining of nucleotide to form polynucleotides, right? Now, how do we join the nucleotide? Children, we saw that it is done by various type of bonds, right? Like, so phosphodiester bond will join various nucleotides to form polynucleotide. Now this bond formation, for the entire process, some energy we required, right? So who provides the energy? Like, because of energy sources needed to provide energy during the replication process, so who provide this energy? So there has to be somebody who will provide all the energy which is needed by the replication. Because these enzymes which we talk about, they can only help in the reaction which is taking place, but they are not to provide any other energy, right? So let us suppose if you go to market, you want to buy stuffs, you want to buy a chocolate, you want to buy a pen, pencil, books, etc. So you need to pay for all of them. While children, you do not earn, means you do not have money. So because you are just a student, you are still studying, so whether you do not have money, but there is somebody who spent for you. Like, from where you get the money? You are going out with your mother will paying for whatever you want to buy. She is the person who provide the energy for purchasing the things, right? So you can think it like that. So in a very similar way, for this replication process, deoxyribonucleoside triphosphate, they can act as substrate and provide energy for this polymerization reaction. Now, what is this deoxyribose nucleoside? You remember nucleoside? We spoke about them. When pentose sugar combine with nitrogen base, they form nucleoside, right? Now when pentose sugar is deoxyribose sugar, when deoxyribose sugar combined with nitrogen base, it forms deoxyribose nucleoside. When this deoxyribose nucleoside combined with a triple phosphate, like how we have ATP, that is adenosine triphosphate, it become a very high energy molecule because high energy is stored in phosphate bond, right? So this is the structure of deoxyribose nucleoside triphosphate. It looks something like that. So it is not only the adenosine, the base can be anything. Suppose in this figure, if the sugar is deoxyribose sugar, right? So let us suppose this picture. In this picture, the sugar is deoxyribose sugar, and this purple color structure is the nitrogen base, which is the adenine. And this portion of yellow color, these are the 3-phosphate group, triphosphate. So this triphosphate stores a lot of energy in its bond, and this energy gets released during hydrolysis, and that is the energy which is being provided during polymerization reaction. Understood? So there are more deoxyribose triphosphate depending upon the nitrogen base which get attached to it. It is not necessary that it has to be adenine. It can be guanine as well. In that case, it becomes deoxyguanosine triphosphate. Same with the cytosine, that it will become deoxycytosine triphosphate. Right? So similarly, it can be thymine triphosphate as well. So depending upon which is the base, the structure will change. And these molecules act as high energy molecules, storing a lot of energy in their triphosphate bonds. And they provide the energy for the entire DNA replication process. So now we are aware with all the machinery and all the enzymes, right? So for this process of replication, of course, we need DNA. Other than DNA, we need quite a few enzymes. And we also need this deoxyribose nucleoside triphosphate to provide energy. So very three important things are there for DNA replication. Let us repeat. First is the DNA, second thing is the enzymes, and third one is the deoxyribose nucleoside triphosphate. So let us now try to understand the exact process of replication. Okay. So how do DNA polymerase works? So now we will understand that working of DNA polymerase and also the process of DNA replication step by step. Now first of all, we have to understand that it is not that the process of replication can start anywhere in DNA, right? That doesn't happen. So there is a specific region in DNA where the process of replication initiates. It is known as origin of replication or the replication origin. Okay, so that particular site where the process of DNA replication initiates, that site is known as origin of replication, right? Now where is exactly it happening? It happens at point from there DNA strand starts separating out from each other. I told you, right? In semi-conservative mode of DNA replication, the two strands of DNA has to separate out, and then copies are created based on each of these strands, which act as template strand. So that is the basic concept, right? So here the region from where strand getting divided into two strands, that point is known as origin of replication, and it does happen anywhere in DNA but on some specific points. Now children, who helps in separating the two strands? That is the very important question, that who actually helps in the separating of two strands. So children, the enzyme helicase, right? It plays a very important role, as we have already discussed. That how does it help in this process? It helps in this process by breaking the hydrogen bonds between the bases, right? So by breaking the hydrogen bonds between the bases, like nitrogen bases, for example adenine and thymine, they are connected with double hydrogen bond, right? Similarly, cytosine connected with guanosine with triple hydrogen bond, right? So all those hydrogen bonds are being broken by this helicase enzyme, right? And when the hydrogen bonds are broken, what happened? The two strands will separate out, and when the two strands separated, a structure is formed. And due to the shape of that structure is known as replication fork, right? So it is said that a replication fork is formed. So once this structure is formed, only then the action of DNA polymerase starts, right? जब replication fork क्रिएट होता है तभी डीएनए पॉलीमरेज का एक्शन स्टार्ट होता है. सो अ स्ट्रक्चर लाइक दिस that is known as replication fork was the original DNA, the parental DNA I mean. So you see that from here it started, it get divided into two halves. Why? Because the enzyme would have come here, right? The helicase enzyme would come here and actually started the breaking of bond in this direction. And as the enzyme keep moving on in this direction, the bonds continuously breaking. As the bond break, two strands separate out from each other, right? In this picture, two strands are anti-parallel to each other. Here you see the anti-parallel polarity which we have discussed. So they are anti-parallel to each other. When enzyme came, it divides the replication fork, it divides the DNA into the replication fork structure. So let us see how the enzyme does this. Children, let us suppose this was your parental DNA, right? This red color and blue color is the parental DNA, where you have two strands of red color and blue color, right? And one of them is from 5 prime to 3 prime and another one is from 3 prime to 5 prime, right? And this sphere represent the helicase enzyme, right? Now as the enzyme helicase move through the DNA, it continuously keep on breaking this hydrogen bond. ये जो green color के जो hydrogen bond आपको दिख रहे हैं, जैसे जैसे हेलिकेज एंजाइम इस डायरेक्शन में मूव करेगा ये हाइड्रोजन बॉन्ड्स को ब्रेक करता जाएगा. एज अ रिजल्ट these red color and blue color strands get separated. So these two strands, one of them is known as leading strand and another one is known as lagging strand. So this one is the leading strand and this one is the lagging strand. ये जो दो अलग अलग हो रहे हैं उनमें से एक लीडिंग स्ट्रैंड कहलाता है और एक लैगिंग स्ट्रैंड कहलाता है. They also anti-parallel to each other. Suppose red strand has 5 prime to 3 prime in this direction. So blue will have 3 prime to 5 prime. अगर रेड वाला 5 प्राइम से 3 प्राइम डायरेक्शन है तो ब्लू वाले की डायरेक्शन 3 प्राइम से 5 प्राइम की ओर होगी, because they are also anti-parallel to each other. Right? Got it? So now what happens next? Once the replication fork structure is formed, now DNA polymerase can do its job. अब जैसे ही replication fork create होता है, now DNA polymerase can do its job. Now, what is the job of DNA polymerase? To create a copy of both these separated strands. DNA polymerase का काम क्या है? DNA polymerase का काम है to create a copy of both these separated strands. ये जो दो strands separate हुए हैं, इन दोनों strands की copy create करना, right? It has to create a copy of this strand as well as this strand. So here we will talk about two type of synthesis. अब यहाँ पे हम two type के synthesis की बात करेंगे. Like is there which is known as continuous synthesis and other one is the discontinuous synthesis. So strands where continuous synthesis takes place is known as leading strand. जिस स्ट्रैंड पे कंटिन्यूअस सिंथेसिस हो रहा है, somewhat here, right? वो कहलाता है लीडिंग स्ट्रैंड और जिस स्ट्रैंड पर डिसकंटिन्यूअस सिंथेसिस होता है वो स्ट्रैंड कहलाता है लैगिंग स्ट्रैंड, right? So please understand the concept carefully now, because here things are not very simple. You need to understand what is happening. What is DNA polymerase doing? It has to create a copy of this strand, right? Now this side the synthesis is continuous. This side the synthesis is continuous. The new strand is being created in a continuous manner, right? अब यहाँ जो synthesis हो रहा है, it occur into the continuous manner. But on the other strand, that is known as lagging strand, the synthesis of the new copy of new strand is discontinuous. So if you see, the arrow are broken from the middle, right? तो अगर आप इसमें यहाँ पे ध्यान से देखोगे तो ये जो arrow है बीच में से टूटे हुए हैं, right? So it is not a synthesis continuously. Breaks are present. Now how and why we'll have those breaks? We will understand कि ये breaks क्यों हैं. So children, first of all, let us talk about the continuous synthesis. So सबसे पहले तो हम continuous synthesis के बारे में बात कर लेते हैं, because it is comparatively simple. Now continuous synthesis happens on the leading strand. अभी मैंने बताया कि जो continuous synthesis होता है ये जिस strand पर होता है वो वाला strand कहलाता है leading strand. Here the DNA polymerase read the leading strand template and act the complementary nucleoside continuously. ये जो DNA polymerase है ये इस leading strand को continuously read करता है और इसकी complementary base pairing से new strand को synthesize करता है. So in this case it is quite simple. DNA polymerase comes to the picture. It will read the sequence of bases on this strand, and depending on that it will keep on creating the new base on this side, and a new strand will be formed. Clear? Let us suppose that the sequence on this strand is A, G, C, T. Okay. Now the DNA polymerase come. It will just move like this, and it moves in this direction. It will lead this letter, right? And will create the corresponding letters on the other side. And that is how the new strand is created. Like the new strand which is created is T, C, G, A. In that way it is created the new strand. So it is very obvious that since from the strand this is the 3' end, this is the 3' end, and this is the 5'. अब ये 3' से 5' की तरह है, तो जो नया स्ट्रैंड बनेगा वो कैसा होगा? 5' से 3 की तरह. ये है 3 से 5, तो जो नया बनेगा वो बनेगा 5 to 3. So what will be this end? This is going to be, because this is new strand which has two complementary to this strand. So therefore they are anti-parallel to each other, right? So this is how the new strand is created, right? So replication fork is also growing in this direction, right? हेलिकेज जो है उस डायरेक्शन में मूव कर रहा है, तो कंटिन्यूस सिंथेसिस भी इस डायरेक्शन में होता है, right? Because enzyme helicase is moving in this direction. Okay, correct? So as the helicase enzyme move in this direction, so the replication fork is gradually increases. जैसे जैसे helicase इधर move करता जाएगा तो replication fork भी इस direction में increase करता जाएगा. So the direction in which replication fork is growing is the same of the direction in which the continuous synthesis takes place on the leading strand. Understood? So we can observe the movement of the two enzymes, helicase and DNA polymerase, which will show that how the direction of replication fork growing. The direction of continuous synthesis is the same, right? And why it is called continuous synthesis? Because the new DNA is being synthesized continuously. That is why it is called continuous synthesis. Now what is left out? Now we have to see that how DNA polymerase will create a new strand on the lagging strand. DNA polymerase ने leading strand पे continuous synthesis से तो नया strand बना लिया, लेकिन leading strand का क्या? वहाँ कैसे बनेगा? So now we will talk about the discontinuous synthesis. Discontinuous synthesis is more complicated than continuous synthesis. It happens on the lagging strand. So which is the lagging strand? So this side is the lagging strand shown in this picture, right? So this is lagging strand. So initially when the process actually discovered for the first time, it is assumed that people thought that on both the strands continuous synthesis takes place. शुरू में माना गया कि दोनों ही स्ट्रैंड में, चाहे वो ये स्ट्रैंड हो चाहे वो ये स्ट्रैंड हो, दोनों में ही continuous synthesis होता है. But later there was a scientist named Okazaki, like, who knew that the two strands of DNA are anti-parallel to each other. So he said that continuous synthesis cannot take place on both the strands. Right? उन्होंने बताया, Okazaki ने, कि दोनों ही strands पे continuous synthesis नहीं होता. As per him, the process of synthesis cannot the same on the both strands. The DNA polymerase works is that, right? What it does, it keep on adding the new sequences of nucleotide to 3' end of the new strand. So basically it will keep on adding nucleotide to 3' end. Okay, 3' end पर nucleotide को add करता है. So in case of leading strand, there is free 3' end. Leading strand पे हमारे पास free end कौन सा है? Red वाला 3'. तो DNA polymerase is able to do its job. अब DNA polymerase जो है वो 3' end पर नए न्यूक्लियोटाइड जोड़ता है. तो लीडिंग स्ट्रैंड पे तो DNA polymerase आया क्योंकि यहाँ पे 3' end फ्री है, तो easily DNA polymerase is able to do its job. But in the case of lagging strand, here the 5' end is free. So 3' end is not free. Where DNA polymerase can add a free nucleotide. जहाँ पे DNA polymerase free nucleotide add कर सके, वो 3' end यहाँ free नहीं है. Because it can add a new nucleotide only to free existing 3' hydroxyl group. Because if you remember the structure of DNA, I said that on 3' end there is the hydroxyl group और 5' end पे phosphate group free होता है. So for DNA polymerase to work, it needs 3' end free. So it is not possible to avail the free 3' end on both the side. लीडिंग हो या लैगिंग हो, दोनों ही जगह 3' end फ्री हो ऐसा नहीं है, क्योंकि एंटी पैरेलल होते हैं. एक जगह 3' end फ्री होगा, एक जगह 5' end फ्री होगा. They both are part of same parental DNA, right? Because they are same part of parental DNA, and two strands are always anti-parallel, so cannot directly add the nucleotide. DNA polymerase cannot directly add the nucleotide. They cannot directly synthesize the way it was doing on the leading strand. तो जैसे leading strand पे काम हो गया continuous, वो lagging strand पर नहीं हो सकता क्योंकि वहाँ पे तो 3' end free ही नहीं है. What can be done in this case? And this were their need for the enzyme primase. अब यहाँ पर ज़रूरत होती है प्राइमेज एंजाइम की. आपको याद है मैंने पिछली वीडियो में भी आपको बताया था कि प्राइमेज एंजाइम का काम होता है एज अ प्राइमर, क्योंकि डीएनए पॉलीमरेज डायरेक्ट इनिशिएट नहीं करता प्रोसेस को, उसको एक प्राइमर की ज़रूरत होती है जो एक आरएनए फ्रेगमेंट होता है, राइट. तो एक प्राइमेज एंजाइम यहाँ पे अब काम करता है. In this case, DNA polymerase cannot initiate the process, so somebody else was needed. याद आ रहा है जो पिछली वीडियो में हम बात कर रहे थे? So somebody else is needed to initiate the process or to provide the 3' end free. Who is going to provide 3' end? Who is going to provide free 3' end? So that was done by the enzyme primase. So I hope you understand the concept. That's why continuous synthesis cannot take place on the lagging strand, because there is no free 3' hydroxyl group. So this is lagging strand, take place in short separate fragment. Now how synthesis is going to happen in this case? What happens actually? The primase enzyme it will create a very short fragment of RNA. And above this RNA, the DNA polymerase will come. It will add the free nucleotide to the free 3' hydroxyl group of that RNA primer. So that is how the process is going to initiate. So enzyme primase initiate the process by creating a small RNA fragment. Now, as soon as this RNA primer is created, a free 3' hydroxyl group is available to the enzyme DNA polymerase. Now, children, DNA polymerase can add free nucleotide to this RNA primer. So, we got the free nucleotide. So, what happens? A small fragment of new DNA is being formed on the lagging strand now. Now, similarly, much small fragments will keep on forming due to the action of primase enzyme, but we need the complete strand of DNA. छोटे छोटे fragment में तो ये create हो जाता है, but we need the complete strand of DNA. We do not want the pieces, so we need something to join these small pieces. So the another enzyme is there, as I said, children. After enzyme primase, DNA polymerase, which extend the RNA primer, adding the free nucleotide. Now the RNA primers are removed and replaced by DNA, and then DNA fragment are joined together by the help of ligase enzyme, जो हमने पिछली वीडियो में भी बात करी थी, राइट? तो ये जो छोटे छोटे एरो आपको दिख रहे हैं, ये छोटे छोटे स्ट्रैंड क्रिएट हो जाते हैं. अब ये पीसेस में, पर हमें पीस नहीं चाहिए. सो दीज स्मॉल डीएनए फ्रैगमेंट आर फॉर्म, दे आर नोन एज ओकाजाकी फ्रैगमेंट. ये जो छोटे छोटे फ्रैगमेंट बन जाते हैं, ये ओकाजाकी फ्रैगमेंट कहलाते हैं, राइट? डीएनए स्ट्रैंड के जो पीसेस होते हैं, फ्रैगमेंट्स होते हैं, ये ओकाजाकी फ्रैगमेंट कहलाते हैं. Why they are termed as Okazaki fragments? Because it was named after the name of scientist who discovered this process, discontinuous synthesis, right? So finally, what happened? These Okazaki fragments are joined by the another enzyme which is known as DNA ligase. Got it, children? I am sure that now you are able to understand that on lagging strand, DNA polymerase cannot directly do the synthesis because there is no 3' hydroxyl end free. So RNA primer are added by primase enzyme. Now RNA are replaced by DNA formed. Then the small pieces, the small fragment of DNA are formed. क्योंकि हमें complete strand चाहिए होता है, pieces में नहीं चाहिए होता है. So these DNA fragment which are termed as Okazaki fragment, they are joined with the help of another enzyme which is known as DNA ligase, and that is how the complete DNA will be formed on this side, right? So we have started today that the replication fork is formed, and one of the strand is leading strand, and on the leading strand there is the continuous synthesis of DNA, and another one is the lagging strand, there is the discontinuous synthesis of DNA. Finally, the Okazaki fragments are formed, which are joined together by the means of another enzyme which is known as DNA ligase, right? So DNA ligase जो है basically RNA fragments को join करेगा, right? So definitely bond बनेंगे. कौन से bond बनेंगे? Yes, phosphodiester bond, right? So research is still in the process to know about the detail of the DNA replication. So children, this is the synthesis cycle of eukaryotes during the cell division. Basically this is the complete process of DNA replication. In the next video we will discuss about the transcription. So I hope that you got every point clearly. Children, still you feel any kind of doubt, any kind of query regarding DNA replication, so just feel free to ask, right? So children, now your job is to revise and